Noch ein bisschen, gewisse Leute sind noch im Waiting Room. Okay. Okay, then I will start this lecture, which is lecture 2, where I will discuss muscular dystrophies and some of the therapies. I actually figured that this lecture will eventually last more than an hour, but we can interrupt or stop, and then we can continue tomorrow, depending on how much time I need and how many questions you are asking. But before I start, I would like to get you the chance to give me feedback, or give us some feedback, or give me some feedback of this first day. I realized that we had some technical issues, but also if you have questions, I mean, it's now the time to maybe ask. Just unmute yourself or raise hands, or
yeah. About the reports.
Yeah.
Well, you're right. What is expected, but just to make it clear with what is mentioned with the result, etc. For example, with the immunostaining, it's clear we have this picture with the different sections. But for example, for the genotyping, where we answer the questions on Google Docs, but we do not have any results like in the other experiments. What is there expected, or how much details do you want?
I don't know in which group you are.
Oh, sorry, group 2.
Okay, yeah, in group 2 you can, for example, discuss, I guess you have been discussing the constructs, no, and then how you would genotype these guys. I mean, that's part of the question in the Google Forms, I agree, but you could, for example, the raptor knockouts and also the TSCs, they're both floxed alleles. You can discuss how you would find out the floxed alleles. You can also basically repeat some of the answers you gave in the Google Forms. It's not a problem. I mean,
okay, so the protocol is more based on the questions we did. So, like, yeah, no.
In this particular case, the genotyping is really the one that where we have, of course, not really given you the bands, but you have, for example, also in the Google Forms, you have some pictures of genotyping, and you can discuss, in this particular case, this is a transgenic, and you would expect to get a band in the AKT, for example, because this is a transgenic. Or is this clear? I mean, it's just, I think this year, when I compare that to previous years, what you really have the chance now, because you have more time, you have a chance to better understand the results you get, because you don't generate themselves, the results, and you don't go through the microscopes. You really have a good basis now to look at these results and also find differences and interpret what you see. I think this is going to be better than it used to be before, because in the years before you were often very busy just getting the pipetting done, and then you were groups of 4, and then one guy did this part of the pipetting, the other person did this part of the pipetting, and at the end communication was a bit difficult, that you sometimes did not understand what you actually did in terms of scientifically what's the question and what's the result and what's the interpretation. I think this will be better this year. At least that's my hope. Is there any feedback from you that what we could improve in the next few days? Okay.
Well,
yes, go ahead.
We had it in one group that we met again to discuss the questions, because somehow it's, well, if you don't know the answer, you cannot yeah, you cannot answer it, obviously.
Yes.
So maybe it's instead of doing the review the next day, just make a shorter summary or wrap up at the end of the session that you really can answer it. I don't know how serious it is when you can't answer one, probably, but
No, it's
that
not
would be
that
one
serious.
chance.
I think for us, it's really the purpose of all this is that we try to figure out whether you understood these experimental parts. And if, let's say, I know the Google Forms, they all require an answer, but if you say you can also type in, I don't know the answer, and then the requirement is settled. I mean, and then the tutor next day realizes that there was a problem. I think it's really for us to learn what your problems are and whether there's a general misunderstanding of the concepts or whether there's just one particular group that has an issue. I mean, for us, this is the tool to try to figure what you got from the concepts. I mean, and you can also, of course, ask the tutor yourself by chat or by email. I didn't understand, can you explain? But maybe it's not a general issue of the entire, of all of you, but it's maybe just a problem of your particular misunderstanding or not understanding certain things. I guess this is what we aim for. And it all depends on your communication. When you ask, we will try to answer. If we don't ask, we don't know that you don't understand, and then we can't answer. That's why I think these Google Forms are a good way to get really a feedback from all of you, and this is why we did this. More? Okay, then I will start with this lecture, and please bear with me because maybe, as I said, it may take a bit longer, but we can stop. I mean, one thing I would also like to mention is that, as you may have seen in the program, that there is this item meet Marcus, and Marcus, that's me. And this is basically the purpose of this is that you can individually make an appointment with me, a Zoom type of appointment where you can discuss questions you have from the blog course, but we could also discuss if you're interested, for example, to do a master's thesis in our group. We could discuss possible projects or whatever. It's really up to you that we can communicate one-on-one. And to do this, I have set up a sign-up sheet also on Google Forms where you then can add your name and also which group you are, and then we can schedule. I will then schedule a meeting, an individual meeting with you by sending you have already the Zoom link, but then just saying, okay, let's talk from then to then, and then we can do this, and you will have this slot here. Each group of you has 30 minutes. This, of course, is not mandatory. It's really up to you to sign up. We can also have some discussions, informal discussions later on. I will send you this link to the sign-up sheet in the Google Forms after the lecture via Adam, and it's also uploaded into this administration folder in the Adam itself. Okay, then let's see. Okay, then I'll start now with the lecture itself, and I will, as I mentioned, I will discuss congenital muscular dystrophies. In particular, I will discuss the disease and also the mouse models we have on this myrosin deficient congenital muscular dystrophy, which is the topic on the one hand of this group 3, and also the topic of the paper you're reading. And I will discuss the pathology briefly and the treatment strategies, and then I will also give you some insights for those people who do not have group 3, what the idea is behind this miniagrin and this laminin nitrogen constructs. And then in the second part, I will also discuss Duchenne muscular dystrophy, because this is the most frequent muscular dystrophy there is. And there are now really also very interesting and to me very exciting treatment strategies to try to deal with this disease. Okay, again, just as a repetition and a reminder, laminins are these heterotrimeric molecules which consist of alpha, beta, and gamma chains. And just to make it again clear, just to everybody, each of these chains are encoded by individual genes. There are 5 genes that encode different laminin alpha chains, and these alpha chains are these chains here, which starts here. This is the amino terminal end, and then it goes through here, and this forms this triple coil region here together with the other beta and gamma chain, and it ends up, this is the C-terminal part of the laminin alpha 1 chain. They, as I said also in the introduction, they combine with different beta and gamma chains to form these heterotrimers. And these are, the nomenclature is as such that the alpha chain is the first digit, beta chain is the second, gamma chain is the third. In other words, a laminin 111 consists of a gamma alpha 1, a beta 1, and a gamma 1 chain, while the laminin 121 consists of alpha 1, beta 2, and gamma 1. And not all of those combinations are also found in vivo, but there are many different combinations. At the end, we end up with about 16 different heterotrimers, which differ in their alpha composition. You see here the alpha 4 is a alpha chain that has a lack of this amino terminal end here, and also of course is different in the C-terminal end. The alpha 3 chain, which is expressed mainly in skin, is also truncated here at the amino terminal end. But there are also isoforms like this alpha 3b chain that is longer. I mean, this is the same gene but just a different splice version basically. Okay, this is a family of genes which are all components of extracellular matrix, and they differ in their tissue distribution. This is also important, and also how they are expressed during development is different. Now this disease where alpha 2 chain is missing, this is called LAMA2 muscular dystrophy, related muscular dystrophy, or MDC1A for muscular dystrophy congenita, because it's a congenital muscular dystrophy 1A. It's, as I said, this is lack of mutations in the gene coding for the alpha 2 chain. And since the alpha 2 is the main laminin alpha chain that is expressed in muscle extracellular matrix and also in the peripheral nerve, it affects, of course, muscle as well as peripheral nerve. It's a very rare autosomal recessive disease. Oh, sorry, I did something. Incidence is quite rare. I mean, there are estimates that there are about 3,000 to 4,000 patients worldwide. In Switzerland, there are about people, or physicians tell us there are about 20 to 30 people who suffer from this laminin alpha 2 deficient muscular dystrophy, but it's still the most prevalent form of a congenital muscular dystrophy, with the exception of Japan. As I mentioned in the other lecture, Japan, there is Fukutin related muscular dystrophy is the most frequent one because they have a founder mutation in this Fukutin gene. It's very severe in most cases, but there are also milder forms, and we can discuss this also in the context then. And also in the paper, they have a mouse model, which is a mouse model for this milder form of LAMA2 deficiency. Now, the next slide shows you the different mutations there are, and you see here this is basically the mouse gene. On the gene, I mean here with its intronic sequences here and the exons, and you see that mutations that cause LAMA2 deficiency or some of this disease, they're scattered along the entire gene. It's really, some of them are nonsense, missense mutations. Some of them affect here the amino terminal end, some affect something more in the C terminal end of the protein. And this is really scattered. Most of these mutations cause the lack of expression of laminin alpha 2. As I said, these are recessive diseases. If you have, usually you have biallelic, I mean heteros, two different mutations in each allele, which then lead to the complete loss of laminin alpha 2, for example. This is, for example, seen here in a biopsy of a patient where we stained with antibodies against laminin alpha 2. And you see this patient here does not have this laminin alpha 2 expressed, but it has, when you use antibodies against laminin beta 1 and gamma 1, which is this one here, they express this, they contain laminins, but they contain another laminin, as I will show you then later. But as a consequence of this lack of laminin alpha 2, the biopsy is really very severely affected. This is the biopsy of this patient. You see there are a lot of mononucleated cells, probably fibrosis is one of the keys here, and also a lot of degeneration, regeneration of the fiber in contrast to the healthy muscle. And you will see sections of a mouse if you are in group 3 that also has lack of laminin alpha 2. Also looks very dystrophic. As I said, you're going to work with mouse models. There is this mouse model we are working with, which is called a DYW mouse. There are many different mouse models, or a few different mouse models. In the paper you have read, there is this DY2J mouse. This DY2J mouse is less severely affected than the DYW mouse. The DYW mouse is basically lacking laminin alpha 2. But this is really quite fortunate and a bit in contrast to the mouse model for Duchenne muscular dystrophy, that this DYW mice, they really reflect also the human disease, that it's really also an early onset disease. You see that very obviously these mice are affected when they are about 2 weeks old. Their body weight is down, the muscle degenerates, they have deformations of the skeleton like this hunchback here, and they also die quite early. It depends a bit on the cohort, and maybe there's also some variation within the mice. Some mice die at the age of 6 weeks and some die only at the age of about 20 weeks, but it's really a good model for the severe form of laminin alpha 2 deficiency. This DYW mouse was generated by homologous recombination by knocking a LAC gene into the LAMA2 locus, and by this interrupting the reading frame of the gene for encoding for laminin alpha 2. This mouse was generated many years ago by a researcher in San Diego, Eva Engwall, and she has then, to confirm that this is really a laminin alpha 2 deficiency that is causing this disease, she then used a transgenic mouse where she expressed the cDNA encoding mouse laminin alpha 2 just in the muscle using a muscle specific promoter. Very similar to what we do, what the mice you are working with that express here this miniagrin and this laminin nitrogen construct. As you can see here, when she does this on this DYW mouse background, she gets really a huge increase in lifespan. They live, in this particular paper that was published already many years ago, more than 20 years ago, these mice survive more than 8 months. They show amelioration of most of the myopathic, the muscle phenotype, but they still have hind limb paralysis, which is an indication that the myelination in the peripheral nerve is not restored in these mice. And can you tell me why why these mice are still hind limb paralyzed? Does anyone have a clue or a suggestion?
Maybe because for the myelination, the development is crucial.
Yes.
It cannot be reversed so easily.
Yes, that's one of the explanations. Another explanation lies here in this transgene expression. Where do we? Yes, sorry.
Only expressed in the muscle and not in the nerve cells, so it can only restore the muscles but not the nerves myelination.
Yes, exactly. This is probably the most important reason. And would you know where you would express then this transgene, where you would need to express the transgene in the peripheral nerve? What do you think? If you think of the cell components that build the nerve, I mean, why are these? It's a progressive disease. They are initially they can walk, but then they lose somehow the hind limb. The hind limbs are paralyzed. Forelimbs are not paralyzed. What do you think? What are the cells that are important for this in this context? And if I say myelination, what are the cells that cause?
The Schwann cells.
Yes, exactly. These are the Schwann cells. These are these. How do you call it? I mean, like in the brain, you have oligodendrocytes, and in the peripheral nerve, you have Schwann cells that form this myelin, which leads to this insulation of the nerve so that the action potential can propagate fast from the motor neuron down to the muscle. Yes, and I mean, but what this tells you, nevertheless, that these mice, if they live longer than 8 months, that the real severity of the disease is caused by the myopathy. I mean, by the disease in the muscle. And it's primarily a muscular dystrophy, but it has also significant contributions by other tissues. And yeah, this might be then actually also important in the context of the mouse group 3 you're working at, because there we also express the transgene actually only in the skeletal muscle. But this was basically proof that this is caused by laminin alpha 2, and as I said, it's mainly a myopathy. The other approach others have taken is to try to replace laminin 211 by laminin 111. This is actually the approach the paper is taking, just not in the same fashion as here, as they have done. But as I mentioned to you, laminin alpha 1 is very similar in terms of its structural similarity to laminin alpha 2. However, laminin alpha 1 is not expressed at all in skeletal muscle. It also is not upregulated in the absence of laminin alpha 2, but it's for example found in kidney. And when you measure binding of laminin alpha 1 to this complex of proteins here, which are important to link these laminins to the plasma membrane of the muscle, it turns out this binding is quite high affinity. Laminin alpha 2 binds with the affinity of about 20 nanomolar. And it also is a long version, has also these amino terminal ends here, like the laminin alpha 2. This is why in 2004, a colleague of mine has also transgenically expressed the cDNA coding for laminin alpha 1. And she did that instead of using the muscle creatine kinase promoter, she used an ubiquitous promoter, which is the so-called CAG promoter, which is a combination of CMV and chick beta-actin. That's why it's called CAG, for C for CMV, AG for actin. And this drives expression of the transgene in all the cells, irrespective of the origin of the cell. And when she does this, then she expresses it, of course, in the muscle, but also in the Schwann cells. And when they do this, they get a very striking effect that they can basically restore the function of the mouse, that the severe muscular dystrophy with this transgene. They, by the way, used a mouse model which is called DY3K, which is very similar to the DYW mouse. It's also generated by homologous recombination by just knocking out the gene encoding for laminin alpha 2, this LAMA2 gene in mice. And you see here they really get a good rescue. However, the trouble with this, of course, is that the cDNA is about 10 kilobase in size, and there is not really a possibility to make small versions of laminin alpha 1 that would eventually fit viral vectors. I will discuss that with you then later. But what you can see in the next slide is the moving of these animals. If I can. Come on. Cannot move it, but it should be possible. Hey, yeah. It doesn't work. Don't ask me why. It worked, but maybe in the presenter mode it doesn't, and I can't activate it. Yeah, anyway, you will see mice that move around that are basically, it's very hard. You have a hard time to distinguish the two mice. One is the wild type mouse, and this other mouse here would be a laminin alpha 2 deficient mouse that also expresses this laminin alpha 1, and you basically don't see much of a phenotype of a difference. These mice also survive at least 2 years. This has been documented. Okay, but the question is also what else is then upregulated in this laminin alpha 2 deficient mice here? As I said, when you have lack of laminin alpha 2, there laminin is still expressed, and one of the laminins that is expressed 411 that instead of laminin alpha 2 has an alpha 4 chain, and there are also other isoforms that are expressed in the skeletal muscle, such as the laminin alpha 5 chain, which is also a longer version. Again, has this amino terminal end here, but they are usually expressed only. Hold on. Marco tells me that maybe I have selected the pointer instead of the arrow for my mouse. That's maybe true. Should I try back? I try back if you have time to see this movie. So now, oh, gosh. Now everything's a bit slow. How can I select? Oh, now it's gone. Maybe it's not worth it, huh? And Joe, now you don't see anything anymore? I will come back. Uh-huh. Now I need to share. Now I'm not sharing anymore now. Now I share again. So now, and Marco was right. Ah, but it's so slow. Ah, do you see it, or does it get stuck maybe?
It's okay.
Okay. Well, you see, it's really, you have a hard time to distinguish the two mice here. And one is a wild, this I think is the wild type, this is the transgenic. Okay, now this is the issue with the expression of these other isoforms. Laminin 411 is expressed mainly at the neuromuscular junction, also in the peripheral nerve, but it's not expressed in the entire muscle extracellular matrix. And laminin alpha 4, which is a full-length version of this, is expressed also at the neuromuscular junction. But this becomes important then now in the context of the mice as well as in the patients, because in the patients, in the patients as well as in the mice, when you stain for the laminin alpha 4 chain, you see that these DYW mice, they start to express now, or you start to see alpha 4 chain expressed not only in the blood vessels and at the neuromuscular junctions, but also now in the entire extracellular matrix surrounding individual muscle fibers. This can also be seen here in the context of when you do a Western, you really see that they express quite a high amount of laminin alpha 4, and when you just measure laminin beta 1 and gamma 1, you see there is no difference really between wild type and DYW mice. This increase in laminin alpha 4 is based on the expression of laminin alpha 4 on transcript. You have an increase of laminin alpha 4 transcripts. This is based on the fact that laminin alpha 4 during development, now when you compare wild type situation, the laminin alpha 4, this is a Western for laminin alpha 4, is highly expressed during development, but then in the wild type it's replaced by laminin alpha 2, while in the DYW mice this laminin alpha 4 remains high throughout adulthood. And this is the reason why why you still have a lot of laminin alpha beta 1 and gamma 1. The same, really that's important of course if you think of therapies, the same is true also for human patients. They also have high amounts of laminin alpha 4. They have absence of laminin alpha 2 of course. And they have also the same amount of beta 1 and gamma 1 chain expressed in their biopsies. And you can again see here when you stain for beta 1, gamma 1, it's also high, and laminin alpha 4 is increased in these patients. Also again, when you measure this, you get an increase in the amount of laminin alpha 4 just as measured in the Western blots here. In other words, humans as well as mice have really also mechanistically very similar compensatory mechanisms, if you wish to say, in the disease. Nevertheless, when you look at the basement membrane and or extracellular matrix, basement membrane is a expression of a structured extracellular matrix. When you visualize the basement membrane here by electron microscopy, you see that basement membrane is an electron-dense structure. This here is the muscle fiber. This is the basement membrane, and you see it's really an electron-dense structure in a wild-type situation, which is this endomysium structure basically. in the case of DYW mice and also in the patients, you see that this basement membrane is disrupted. It's really like interrupted. It doesn't really have this nice high density structure as you see in a wild type situation. And this is symbolized here. And the reason for this is that laminin 411 lacks two important, and that's what we believe at least, lacks two important domains. On the one hand, it lacks a very strong interaction with its receptors on the cell surface of muscle fibers, which are on the one hand integrins and most importantly alpha-dystroglycan, which is this protein here. And when you measure just in vitro binding of laminin 411, a purified laminin 411, to either integrins or alpha-dystroglycan, you see that this does not at all bind. Here is relative binding here, while laminin 111 or 211 for this as well would bind with very, very high affinity to integrins as well as to alpha-dystroglycan. And then one argument could be that if you insert now a linker protein, that's what we did with this miniagrin, that confers binding of laminin 411 to alpha-dystroglycan, you could eventually restore this connection of the extracellular matrix or the basement membrane to the underlying plasma membrane. And for this, we generated out of this big protein agrin, where we have seen that it binds via its amino terminal end to laminins into this coiled region here, and on the other hand it binds to alpha-dystroglycan via its carboxy terminal end here. We made a fusion construct of which we called miniagrin, which when you add that now to 411 in culture, this is then you see that the binding now to alpha-dystroglycan also strongly increases, similarly to what you see when you add laminin 111 to this in this assay. In other words, you can really bind now laminin 411 now binds to the surface of these cells. The other issue with 411 is that it lacks domains that are important for laminin polymerization. Laminin polymerization is basically the formation of these trimers here via the amino terminal ends of the long laminins. I mean, you basically require an amino terminal end of the alpha chain together with a beta chain amino terminal end and the amino terminal end of the gamma chain, and then they form these trimers here and form a tight network in the muscle basement membrane. And this amino terminal end is lacking in the alpha 4 chain. And again, when you measure polymerization of laminins, and you do that with laminin 411, and you measure the extent of polymers, there is no polymer formation with laminin 411. While if you do that with something like laminin 111 or 211, you get this polymer formation. This is why we generated yet another linker protein. This linker protein consists of a fusion of an LN domain, which is this domain here of laminin alpha 1, which is this here. This is alpha 1, just the protein structure. And we fuse this to a binding site that confers laminin binding to the gamma 1 chain, which is another protein of the extracellular matrix called nitrogen. And we basically fuse this LN domain to this laminin binding domain of nitrogen, within the hope that with this now we can present another LN domain, another amino terminal end, similar to what happens with laminin 211. And indeed, when we now add laminin 411 at this construct, which we called alpha LN nitrogen or alpha LNND, you also see that now laminin 411 forms these polymers, which would argue that now you also have again made laminin 411 like a polymerizing laminin. And of course, this is all in vitro. You want to test this also in vivo. And for this, we generated these mice you deal with. These are these DYW mice. On the one hand, miniagrin is included, and then we also made mice that express both this alpha LNND and miniagrin or MAG. And these are the mice you also have in your cohort. We express these transgenes again with a muscle-specific promoter. In other words, we are not expecting to restore any of the peripheral neuropathy, but we hope to restore the muscle histology and the muscle function in these animals.
Yeah. I have a question about the laminin 411.
Yeah.
Is there a beneficial effect of the upregulation in DYW mice where there are no linkoproteins?
Yes, I mean, if you can do the experiment where you knock out laminin alpha 2 as well as laminin alpha 4, and these mice, they don't make it. They really die shortly after birth. I mean, it has some compensatory effect, laminin 411, but it's not sufficient. You could now also ask why is not laminin alpha 1 upregulated, because that would do the job, but basically the answer is I don't know. It simply is not. Maybe the promoters are wrong or different. Yeah, but it's a good question. Thank you. Are there more questions? Okay. Okay, then when we do this transgenic experiment and we just look at the basement membrane structure again and we compare again DYW with wild type, where you see here this nicely structured basement membrane, you can obviously see that these intermediates here expressing a single transgene have already some effect, but you can see here in the double transgenics that this looks really quite similar, if not identical. You actually have a hard time to distinguish this from a wild type situation here. I should also say we express, of course, these transgenes already in embryo. I mean, these animals, they are really never as sick as they can get, which is, of course, different if you want to treat patients. Okay, what we then also see that we get a really strong restoration of muscle histology. You will see this as well. You will do H&E stainings, or you will see H&E stainings and serious red stainings, and you will also see that we get this improvement in contrast to the DYW mice. If you do fiber size distribution, which you also do in your mice, you will also see that the double transgenics, they really look quite, they get in the right direction like controls. They may not really get the same fiber size distribution like the control, but they're getting close to this. When you measure fiber number, they are quite similar to the control. When you measure fibrosis, they're also quite similar to the control. I mean, the DYW mice have a lot of fibrosis, which you can measure by basically measuring collagen content in the muscle, by measuring hydroxyproline, which is a modification of proline which only is found in collagens. And most importantly, of course, this is what we observe. This is basically what you see here. When you compare the survival of a DYW mouse, in this particular cohort, the median survival of this DYW mice was about 15 weeks. In these double transgenics, the median survival is 81 weeks, and we actually have some mice that these double transgenics, they become more than 2.5 years old. The oldest one we had, and then actually sacrificed it because we needed to look at some of the muscle at this high age, was 2 years and 7.5 months. And I should emphasize that this, as I said, because we express the transgenes only in the muscle, these mice, they still have a very severe hind limb paralysis. They actually cannot move their hind limbs anymore at this particular age, and they just walk in the cage with their forelimbs, and pull them across. But they still, survival is not really affected by this. I should also say, of course, if you can imagine, animal experimentation, that people who don't like animal experimentation, they also don't like to see mice paralyzed in their hind limbs. And we had a hard time to convince the animal vets that we can actually do these survival curves, because this is really important if you think of translating that to patients, because we basically needed to show them that these mice are really not suffering that much. I mean, it's very similar to someone, a human, who injures his spinal cord and then is paralyzed. I mean, these people also don't suffer. I mean, of course they suffer, but they don't suffer that you need to euthanize them. I mean, in the animal research, I find this really strange that you basically say if you think an animal suffers, you euthanize the mice. But while, I mean, in the human situation, you cannot imagine. I mean, it just doesn't make any sense to me. But anyway, this tells you that this mice really make it despite having this hind limb paralysis. Okay, but what this basically shows is that these transgenes, they can restore the muscle basement membrane. They also restore many of the parameters that are important for survival in the muscle. That's why I think it's a really good concept, proof of principle, that using these linkers could eventually be a means also to treat patients. And it's also really, I think, a nice demonstration that the muscular dystrophy in these mice is really a problem in the basement membrane assembly on the one hand, and its connection to the sarcolemma. And this is the two functions we restore in those animals. Now, if you think of this scheme here, what I told you before, that we have these structural defects, which are the starting point of having this fall down from instability to muscle loss here. And I also mentioned in the introductory lectures that you can interfere always at different levels by different means. And as I showed you, laminin alpha-1 overexpression gives this 2-year survival. And with our transgenics, we are actually also very close to this situation here. We get survivals of more than 2 years even. While if you use other approaches that are listed here, they are not that efficient as if you interfere here with the structural defects. But as I mentioned also, these are more generally applicable in the context. And I should also mention that inhibition of apoptosis, this is the first clinical trial that was made in these particular patients deficient of laminin alpha-2, where there was a phase 1 trial was made, just a safety trial, which was based on these effects here, that inhibition of apoptosis is also something that improves survival in these mouse models here. However, efficacy in this phase 1 trial was not really observed. Okay, with this, do you have questions to this, or? Okay, then I will go on and introduce this other disease, Duchenne muscular dystrophy, which, as I said, is the most frequent one that is found, and it's really because it's a quite frequent muscular dystrophy. It's also where most of the treatment strategies have been developed, and I want to discuss with you two strategies. One of the strategies is gene therapy, and the other strategy is exon skipping, because I think these are really both based on a fundamental understanding of the disease pathomechanisms that eventually also give exciting prospects for eventually treating those patients. Okay, Duchenne muscular dystrophy is a very old disease. I mean, it was discovered by a physician called Duchenne in more than about 140 years ago, and he called it paralysis, a hypertrophic paralysis of children that is caused by a problem in the brain. And it calls it paralysis with a pseudo hypertrophy. And you see this pseudo hypertrophy here in the calf muscle, because this seems to be an effect of, on the one hand, regeneration that is progressive, and on the other hand by fat tissue that infiltrates this muscle. It's genetically determined. That was already clear quite early. It's very progressive, and the primary defect lays in the muscle fibers. Then it took more than 100 years until the gene underlying this disease was identified. The gene was then called dystrophin, and it was, I should emphasize, you were all not born at that time probably. This was the first gene that was identified underlying a genetic disease. And we are now 30-something years later, and we have now a panoply of genes that are associated with genetic diseases. It's unbelievable the progress we make on this. And upon the identification of this dystrophin, people were speculating, now we know the gene. Now we can actually treat the disease by just bringing back the gene to these patients. And this, now 30 plus years later, is still not really possible, but we are, I hope, on a good way to actually be able to do this. And I want to discuss that with you. Duchenne muscular dystrophy affects, is one of these diseases that affects limb muscles. The patients, they suffer from loss of ambulation at some time. It affects this complex that is also affected in the laminin alpha 2 deficiency, but it affects this protein here, which is a very big protein that links this protein complex here to the actin cytoskeleton. It's an X-linked mutation. It's localized in the X chromosome at localization P21. Therefore, it affects only boys. It's the largest gene in the human genome. It's 2.6 megabases in size, encodes many exons. Its synthesis of this primary transcript takes 16 hours. The mature mRNA is 14 kilobases. It's expressed in muscle, brain, and the protein is also of huge size. In Duchenne muscular dystrophy, there is no protein expressed. And often there are also these mutations can be scattered all over the gene because it's so big. There are point mutations that eventually then cause frame shifts, but there are also deletions. The clinical features are a delay in walking, clumsiness, and so on and so forth. It also affects actually the heart. But what I would like to show you here is a so-called Gowers maneuver. This is how these boys get off the floor because they're weak. They need to a particular maneuver to get off the floor, and this is shown in this movie here. It goes on the knees and then basically uses the arms the hands to get up. This is called Gowers maneuver. As I said, this is also a progressive disease. When you look at 2 years, you start to see some disease course. You see here many of these fibers are roundish. There is also some connective tissue here. They are of different size. Some are very small, some are quite large. When you then go on, you start to see a bigger heterogeneity in the disease, more of these necrotic fibers, but also some fibrosis. At the higher age now, you start to see invasion of adipocytes, fat, into the muscle because the muscle is going to be replaced by fat tissue. And these boys, they often cannot walk around the age of about 10 to 15 years. It depends a bit. These boys eventually also die because of respiratory failure. It used to be that they die at the age of about 20. Now I think they survive much longer because we have all these kind of treatments that, I mean, palliative treatment, for example, overnight ventilation, that prevents them from dying because of respiratory failure. When you do cross sections through a patient through a biopsy, and you stain for dystrophin. Dystrophin is stained very homogeneously and along all of the muscle fibers, as you can see here. If you stain with antibodies against dystrophin a Duchenne patient, you see basically no expression whatsoever of the protein, also seen here in the control. But once in a while, you get these dystrophin positive fibers, which are called revertant fibers. Does any one of you have an idea why you get these dystrophin positive fibers here in a muscle section of a Duchenne patient? In this particular case, there was exon 50 was deleted. Why could be in the soma be all of a sudden a cell be positive of dystrophin, and think it's just one copy? And what can happen to somatic cells? If you think of cancer, why does
Yeah. Maybe that the mutation is reversed.
Yes. There could be, it's a very good idea. I mean, there could be a second mutation that, for example, if this exon 50 is deleted, it may be causing a frame shift here, and then the loss of dystrophin, because it becomes also unstable, the message. And if you have yet another mutation which would also cause a frame shift, another frame shift, so that the protein again goes into the frame, then you could eventually restore this. And this observation of these revertant fibers and your interpretation was really also the starting point of trying to figure what could be ways to treat this very severe disease. Because you saw that you can manipulate somatically a bit the frame or something, and then you eventually will get again dystrophin positive fibers. Very good. Another example is Becker muscular dystrophy, and this particular patient I think is really famous because what this patient has is a huge deletion inside here in this particular region. If you use antibodies against the amino terminal end, you get dystrophin fibers. If you use antibodies in this middle region here, against this middle region, there's no staining whatsoever. If you use antibodies against the carboxy terminal end, you again get dystrophin positive fibers. When you do Western, the patient is dystrophin positive, but his dystrophin is much smaller than the wild type dystrophin. And importantly, this patient really has a deletion of exons 14 to 41 and has really a much shorter protein. But what is really important is the phenotype of this patient is not at all as severe as in a Duchenne boy. This person is maybe 60 years old. He can walk on his toes, which is someone cannot do if he has a very severe muscular dystrophy. Has some weakness to walk up the stairs. Has also this hypertrophy of the calf muscle. But importantly, still walks. Has Gower's maneuver to get up. but can lift the arms also with no, I mean, a Duchenne patient would never be able to do this at the age of about 10 years. And this was also another important observation, because that triggered the idea that you could eventually transform a Duchenne patient into a Becker patient by basically making Duchenne into Becker by making smaller versions of the dystrophin protein. And this was really also the starting point to eventually try then gene therapy to generate smaller versions of dystrophin, and basically showing that they're still functional, because this gene therapy has certain requirements, and dystrophin, full-length dystrophin, would never really fit into a gene therapeutic approach. Now, before I stop, I just want to briefly show you these gene therapy approaches, because I find this quite exciting, and it's really, since I'm so long in this business, gene therapy was always thought not to be working for quite some time. I mean, 10 years ago, people would laugh at you when you would say, "Yeah, one should eventually try to do gene therapy to treat those patients." But this really has changed dramatically nowadays. Okay, gene therapy, what this basically is a therapeutic delivery of nucleic acids into a patient cell as a drug to treat disease. That's the definition of gene therapy. And you may have heard of this, and maybe you have watched, what was it, Rundschau, I think, about 2 weeks ago, that there is a gene therapy now available on the market. This gene therapy is sold by Novartis, was approved by FDA, the American disease agency, and it is used to treat spinal muscular atrophy. It's a gene therapy that brings back the gene affected in spinal muscular atrophy. It's treating very young children. They would die otherwise in the first 2 years of their life. They bring their gene therapy back. It costs a fortune. It costs more than $2 million. And this has also triggered the discussion as to how much you can ask patients to pay for such a treatment. But nevertheless, I think for me important is that there is really the first proof of principle that gene therapy can work and can be approved for a neuromuscular disease. The essential components for gene therapy are, on the one hand, the vector, which is a virus usually that delivers the transgene to target the cells with minimal adverse effects. And then, of course, the other essence is the transgene that produces then the functional version of the protein of interest. And thirdly, you also have to decide on a promoter, because this drives then the expression in the intended tissue. And for the vector, it's now very clear that adeno-associated virus is the way to go, because this is a non-toxic virus, as you can see here. This is actually a single-stranded RNA virus that infects many tissues, depends on its capsid, but we have now generated many different recombinant viruses that affect many tissues. The AAV genome is also important, is replaceable. We can replace this, except some of the essential components like the inverted terminal repeats and things that you need to generate the RNA, DNA. It's replication deficient. You can basically assemble all this outside in a dish. It also does not incorporate, or very little incorporates into the host genome. This is important because if it remains episomal, it cannot cause cancer, because one of the issues could be that if you integrate randomly into the genome, you can of course also cause mutations, somatic mutations, and if it happens that your gene of interest incorporates into a tumor suppressor gene, then you eventually will trigger cancer. It has a broad tissue tropism and a high transduction efficacy. That's important. It also has been shown to at least stay around for at least 10 years in the muscle. And the wild type AAV is not at all associated with any human disease. The big drawback of this vector is that it has quite a small cloning capacity. Your transgenes can only be about 5 kilobases in size, and with dystrophin being 14 kilobases, there is no way that this would even fit into AAV. But what this basically shows here is that the virus is incorporated into the cells. Then the single-stranded AAV RNA is released. It's second strand synthesis, and then it's basically episomally maintained. Okay, the other thing, as I said, the transgene, you have to select for the transgene and the promoter. And for dystrophin, as I mentioned, this is a huge protein of 400 kilodalton and plus, and 14 kilobases the cDNA. But however, there are now versions, and this is based on the Becker experience, that you can really get rid of many of these rod-like domains here, and you still get something that is functionally quite similar, which they ended up making a 3.6 kilobases sized dystrophin. The cDNA is only 3.6 kilobases, and this now fits into AAV. And again, just to tell you, of course, this took many years, but one of the key issues was also to use then mice to test this. And this is a paper which was published now 16 years ago in Nature Medicine, where they injected these AAVs into mouse models for dystrophin deficiency, which are called MDX mice. And this is basically a staining for dystrophin in a wild type situation in the MDX mice. Again, you also see these revertant fibers. And when you now inject 10 to the 12 vector genomes into a mouse, into the tail vein systemically, you see that you get many fibers now positive. If you increase your vector load by the factor of 10, you get quite a homogeneous expression of this microdystrophin, and this also has improvements of the force you generate. These are the wild types, the MDX mice, and these are the transgenic mice. You see that there is an improvement in the force you generate here in these muscles when you test them. But this has led now to the use of this kind of concept, and there are now at the moment, as we speak, 3 different companies running clinical trials that use now AAV that expresses microdystrophin. And some of them are really quite promising and quite far-fetched. I just want to show you a few of a company presentation, which is a company called Sarepta, that has presented this a year ago. And they have treated, at that time, they had treated 4 boys that were treated with this AAV. I mean, you inject that into the bloodstream, and then you have to manage somehow the initial reaction to the AAVs. But then it looks quite safe at the moment, and then they stained basically for dystrophin positivity in a biopsy of their muscles, and you can clearly see here that you get quite an homogeneous and strong expression of this microdystrophin in the different subjects. You can also measure the expression of this microdystrophin in Western blots in these biopsies, and you can see here that they estimate that their the amount of microdystrophin expressed in those patients is quite similar to what you get in a a normal situation. Although I must say this is quite difficult to justify, because I mean, if you think of plotting efficacy of a big protein and plotting efficacy of a smaller protein, it's very difficult to estimate really how much of this microdystrophin you generate here in these injected humans. And they estimate they get about 3 copies of this dystrophin per nucleus in a muscle fiber, which is also very high, but I'm not sure how reliable this is. But what they also see, and this is of course important, they see really an improvement of the motor skills of these boys. I mean, what you see here are these 4 patients, their baseline score, and this score is based on a document called North Star Ambulatory Assessment of neuromuscular diseases, and you get certain scores. I have uploaded this, you can look at this if you're interested. And then they see after injection, day 30 until day 270, they get a clear improvement in these scores in most of the cases, except in this patient it's quite weak, but at least it's a stabilization. If you think that it would be a progressive disease, you would expect that the scoring would go down with age. But overall, it looks as if this would be beneficial for this. Okay, and I think with this I would like to stop, and then next tomorrow I will then finish with the rest. Are there questions for the moment?
Yes, I have a question about Duchenne patients.
Mm-hmm.
What happens if you inject steroids?
Yeah, actually, this is standard care. That they are now all of the Duchenne patients are treated with steroids. The mechanism is not really 100% understood, but one could think it's an anti-inflammatory response. But what these patients, it has really been shown that if you treat Duchenne patients at a young age and they're still ambulatory, they are still capable of walking for at least 2 or 3 years longer than if they are not treated. Steroids have some side effects. They make, for example, a short stature. They don't grow anymore, these boys. And they also they increase in weight. This is the other problem. Another company has developed a steroid-like compound that seems to have lost these side effects of the steroids, but still has the beneficial effects of the steroids on Duchenne. They're currently running now clinical trials with this. Maybe that would be another thing that would help. But of course, steroids don't cure the disease. It's just they slow down the progression of the disease. But it's now standard of care. I mean, everybody all the physicians who treat Duchenne patients recommend to treat the boys with steroids, although some parents don't want to have it because of the side effects.
Thanks.
Other questions? Okay. If this is not the case, I wish you good appetite, have a nice break, and I hope you will be strong again to continue in the afternoon.